In this module, I would like to talk about another type of coat protein called COP1, and I would also like to talk about mechanisms of how vesicles fuse with one another. This doesn't happen at random. Vesicles will only fuse with the other compartment of the cell if they're targeted to that compartment. So let's see, first of all, the mechanism of that. After transport, vesicles have budded off from an ER exit site, they shed their coat and then they begin to fuse with one another. This fusion of membranes from same compartment is called homotypic fusion. It is to distinguish it from heterotypic fusion, in which membrane from one compartment fuses with the membrane of a different compartment. So how do vesicles know which vesicle to fuse with or which vesicle not to fuse with? There are specific proteins, just as proteins have labels on their surface which target them to different organelles, vesicles also have an address or a label on them. This address basically is in the form of specific proteins, and in this case, these are called the snare proteins. So here is a vesicle with these snare proteins. One is shown in red and the other is shown in blue. So these snare proteins, they bind each other. The blues bind the red and the red binds the blue. So I have to tell you here that there are many different types of snare proteins, of course, which help target specific vesicles to specific target sites. And these T snares and V snares basically help vesicles maintain the specificity of their target. So T snares basically is the target snare. This is the target vesicle or target organelle. And in this case, actually, since this is homotypic fusion, so both these T snares and V snares are present on the same vesicle. But just to clarify, T snares is the target snare and V snare is the vesicle snare. So these two snares will fuse with one another. Once this happens, they fuse with another, they sort of twist on each other. Once they twist upon each other, it helps generate enough force to fuse these two vesicles. Now remember that there is water in between these two vesicles that is going to prevent, that is going to resist the fusion of these two vesicles. So the force generated by twisting of these snare molecules helps these two vesicles fuse with one another. As with heterotypic fusion, homotypic fusion requires a set of matching snares. The interaction is symmetrical, with V snares and T snares contributing in the fusion of both membranes. The structures formed when ER-dried vesicles fuse with one another are called vesicular tubular clusters. So when the vesicles bud off from the ER, they start immediately shed their coats and they start fusing with each other. And these fused vesicles are referred to as vesicular tubular clusters. I'll show you a diagram of that too, but I would like to tell you that these clusters constitute a new compartment that is distinct from endoplasmic reticulum, since it lacks many proteins endoplasmic reticulum has, and it is different from Golgi apparatus because it contains certain proteins that Golgi apparatus does not have, and vesicular tubular clusters, they have those proteins. So let's look at that. Here is a graphic representation of what I've just talked about. So here's the endoplasmic reticulum. COP2 coats, they start assembling on specific endoplasmic reticulum exit sites, and they result in formation of these vesicles. As soon as these vesicles bud off, this coat is disassembled, and now we have membrane of the vesicle, out of which specific proteins are sticking out. These are snare proteins. They help these vesicles fuse with one another, and once these vesicles fuse, they form a larger structure which is referred as vesicular tubular cluster. As soon that happens, there's another type of protein called COP1 protein that starts assembling on the surface of vesicular tubular clusters, and just like COP2 helps recruit specific proteins destined for Golgi apparatus, COP1 proteins help assemble proteins which are resident of ER or which are supposed to be in ER. So basically these vesicles carry proteins, ER proteins, back from vesicular tubular cluster back to the endoplasmic reticulum. These COP1 coated vesicles also different regions of Golgi apparatus. So basically what they are doing is they are grabbing the proteins which escaped the system and ended up in Golgi apparatus. However, they are actually required in the endoplasmic reticulum. So these proteins are captured with the help of COP1 coated vesicles and sent back to the endoplasmic reticulum. Here is an electron micrograph of vesicular tubular cluster, you can see. So how do these COP1 proteins, coat proteins, know which proteins to grab and send back to ER? That would be topic of our next module.